Welcome to another edition of CCSD Living Environment. Today we're going to talk about the chemical components of DNA. So the first question we need to answer is, what is DNA? DNA stands for deoxyribonucleic acid. Remember, DNA is a polymer, which is a large molecule, made up of the same repeating subunit, or monomer, and in this case for DNA, that monomer is called a nucleotide. So the DNA polymer is made up of a nucleotide. Nucleotides have three parts. The first one being a 5-carbon sugar called deoxyribose. Remember, sugars end in ose. A phosphate group, and one of 4 nitrogenous bases. So our options for nitrogenous bases, it could have adenine, or A, which is considered a purine, guanine, or G, which is also considered a purine, cytosine, or C, which is considered a pyrimidine, and thymine, or T, which is also a pyrimidine. In your note packet, it gives you nice pictures of purines and pyrimidines. So the purine molecule is a larger molecule, which would be our A and G. The pyrimidine molecule is a smaller molecule, which would be our C and T. Nucleotides can be represented simply by shape. So generally speaking, these are the shapes that we hook together to represent a nucleotide. The circle would be the phosphate group. The pentagon would be the sugar, which remember is deoxyribose. And that rectangle is going to be our nitrogenous base, which can be represented as A, C, T, or G. So this whole structure of DNA, we know that now. You're familiar with that now, but there's a history to that. So we need to look at how they discovered the structure of the DNA molecule, and we're going to go back to 1950, which, yes, seems like a long time ago, but my dad was born in 1950. So all this stuff we're doing with the history of DNA really wasn't that long ago. In 1950, a man by the name of Edwin Chargaff came up with this idea, and he determined that the amount of adenine, or A, was always equal to the amount of thymine, or T, and the amount of cytosine, or C, is always equal to the amount of guanine, or G. So A always goes with T, C always goes with G. Well, what does that mean? If A made up 20% of the total molecule, then that would tell us that T also made up 20%. So the 20% and 20% would give us 40% of the total. If I subtract that from 100%, I'm left with 60% between C and G, which would mean that C had to be 30 and G had to be 30% of the total molecule. So if you know the percentage of one, you know the percentages of all of them. This was a huge step in helping to determine the actual structure of the DNA molecule. And the next big piece of information came in 1952 by a man named Maurice Wilkins and a lady named Rosalind Franklin. They did this process called X-ray crystallography, which basically allowed them to take an X-ray, but not of like bones and people like we're familiar with, but more of a molecule. So they actually took a picture of the DNA molecule, and you can see a copy of that picture there in your note packet. So what that picture told them was that DNA had to have this tightly coiled shape, and that's why you see that X pattern. That's how they knew that there had to be that spiral shape that we're familiar with when it comes to the DNA molecule. And all of this led to, in 1953, James Watson and Francis Crick realized that DNA molecule was too thick to be single-stranded, so it had to be 2 strands wrapped around each other. And that Watson and Crick model said that there are 2 complementary, complement because A with T, C with G, strands of nucleotides in a ladder type organization. So I drew a ladder like what we're familiar with when we think about a ladder, but remember, the picture told us that it was twisted. It's like a spiral staircase. So take a regular ladder and twist it. And so what you end up with is a spiral staircase. This is the structure of the DNA molecule. So these three pieces together, the fact that A is always with T and C is always with G, the picture to say, hey, there's got to be a spiral shape to this molecule, and then finally, it was actually February 28th, 1953, that James Watson and Francis Crick said, okay, we got this thing figured out. And it took them a long time to try to cut out those molecules because they actually wanted to show how the nucleotides fit together. So it was some trial and error, and finally Watson and Crick came up with the structure of the DNA molecule that we are familiar with today. So let's talk about that ladder. The sides, or the uprights of the ladder, are alternating phosphate and sugar molecules. So it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And they're connected by what we call a phosphodiester bond. So the bond that holds the phosphate of one to the sugar of another is called a phosphodiester bond. The bases then are attached to opposite sides of the ladder in the middle. The bases make up the rungs of the ladder. Those are held together by weak hydrogen bonds. So just think about that for a minute. We've got these phosphodiester bonds, and those are pretty strong bonds, okay? And then they're telling you that there's weak hydrogen bonds holding it together in the middle. Finally, we're going to come down to the complementary base pair rule. Only two base pair combinations in the DNA molecule are possible. A with T, C with G. It's always adenine and thymine, cytosine and guanine. Now, A with T is the same as T with A. It's like 2 plus 4 or 4 plus 2. It's the same thing, but those are the complements. They're always together. C with G or G with C. Either way, 3 plus 4 or 4 plus 3. It can go either way, but it's always A and T and C and G. So if you need a little mnemonic to help, all turtles can gurgle. I don't know if it's true or false, but we've got AT, CG. So we find DNA in a double helix, meaning the structure as a whole is twisted to form that spiral structure. So remember, think about a ladder, a regular ladder, and then twist that ladder. And when you twist that ladder, you get a spiral staircase. So we're going to take that phosphate sugar upright with the bases in the middle, and we're going to twist that to give us a double helix. So if we look at it, phosphate, sugar, 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 phosphate, sugar. And my bases are connected in the middle. A is always with T, C is always with G. And again, it doesn't matter if it's TA, AT, GC, or CG. The two strands are complementary and anti-parallel. So within this picture, what shows the complementarity of the molecule? And that goes back to A with T, G with C. So the complementarity is in the base pairs. What about that picture that I've drawn here and that you have in your notes shows that it is anti-parallel? Well, we know that parallel means that they're running the same way and those lines aren't going to touch. Well, these things are running the same direction. They're not going to touch. But what makes them anti-parallel? And it's because the phosphate group is up here and the phosphate group is down here. So that means then that the sugar molecule is going to be down here and the sugar molecule is going to be up over here. So because of the fact that the two molecules are running in the opposite direction, we say that they are anti-parallel to each other. So with those two strands, I have complementarity in the base pairs and an anti-parallel nature in which direction the sugar and phosphate backbone is going. So if we took this molecule and we twisted it, that would be our double helix. That takes us through and talks simply about the structure and the chemical components of the DNA molecule. Next time we're going to look at DNA replication, which obviously is far more important to the way life works.